Okay, so chapter 10 focuses on muscle tissue, and I just want you to recall that the four main types of tissue found in the body are, of course, muscle tissue, connective tissue, epithelial tissue, and then nervous tissue. Since we're focusing on muscle tissue, predominantly on skeletal tissue, I want you to recall its general function. You probably learned this in lab. Muscle tissue's general function is to shorten along its long axis. And to sort of picture this, think about shredding chicken. And its long axis is, for instance, along the fibers of whenever you shred chicken. That's its long axis. And muscle cells, since they shorten along its long axis, and if you want something to move, you need to, or if you want something to change shape in the body, you need to put muscle tissue inside of those structures. Okay, so let's go over some of these types of muscle tissue and general characteristics of these types of muscle tissue. Skeletal muscle, that cell sort of looks like this. It's a long cell with multiple nuclei. The nuclei are pushed out to the sides of the cell. It is striated, meaning if you looked at this muscle tissue underneath a microscope, you could actually see stripes. Okay? And it is also voluntary, meaning we think to make it short. We think to move or contract a muscle cell, the skeletal muscle cell. Whereas with a smooth muscle cell, it looks sort of like this. It's spindle shaped. It has one nucleus per cell. It is non-striated, meaning we do not see these stripes. And is involuntary, meaning we do not think to contract a smooth muscle cell. So we would have these in places like smooth muscle around our digestive tract and lining our or around our blood vessels. Now, cardiac muscle tissue is even more different, and it looks sort of like the branches of a tree. So this would be one muscle cell, this would be another one, and they're connected by these special lines here, and they have a name. There's typically only one nucleus per cell, and they are striated. So they're striated, let's write all this down, striated, one nucleus per cell. They're branched. And connected by these special structures called intercalated discs. It helps one muscle cell communicate with the next muscle cell. And so if you think about this, do we think to contract our heart, which is made up of cardiac muscle tissue? And the answer to that would be no, so we would deem this involuntary. Okay. So most skeletal muscle, most attached to bones and span joints. So we can shorten this muscle, skeletal muscle, and cause movement. For this to happen, you need at least two attachment points. So remember, a muscle action can be determined by the attachment and the fiber direction, or bundles of the fascicles. And we said we need at least two attachment points, and we call those origin and insertion. So if you remember from lab what the difference between these two are, the origin is the less movable region, and the insertion is the more movable region. And these muscles typically come in pairs. movement pairs. So like one muscle in a pair causes abduction while the other causes adduction. And we call these antagonistic pairs. Muscles can't relax themselves and they need their antagonistic pair to relax it back out. I also have listed here some terms and names we need to be familiar with. The first three are found in other types of cells but with different names. So sarcolemma is basically a cell membrane with some other extra stuff added in. Sarcoplasm is basically the cytoplasm of a muscle cell. And the sarcoplasmic reticulum is the smooth endoplasmic reticulum of a muscle cell. It basically, in the muscle cell, it basically wraps around these bundles of myofibrils. And myofibrils are bundles of contractile proteins. They're the contractile proteins in a muscle cell. If you wanted to compare it to something in a normal cell or a typical cell, it would be sort of like the cytoskeleton of a cell. The other term is a T-tubule, and it's sort of like a funnel down into the cell that helps to carry charge. It helps to carry or distribute charge into the muscle cell. Okay. So here we have a picture showing each level of the muscle tissue, and there are certain things that we need to know here. So pick a good color here. We have the bone and the muscle attached to the bone, and what's attaching the muscle to the bone is the tendon. Tendons attach muscle to bone, and an extension of the tendon surrounds the muscle here, and it's a bundle of these fasciculales or fascicles. So this whole thing right here is considered the muscle organ. And a muscle organ, an example of that would be like the deltoid or the pectoralis major. That would be considered the muscle organ because we have more than one type of tissue here. Okay, so now this muscle is made up of bundles of fascicles, and the bundles of fascicles, if we pull out one of those fascicles, it's also surrounded by a tissue covering, a connective tissue covering. So they have their connective tissue here in blue. So the first one covering the muscle organ, which is a continuation of the tendon, is called the epimysium. And then the one surrounding the bundles of fascicles here, that would be the perimysium. And now if we take these bundles of fascicles out here and pull an individual one out right here, that's also surrounded by a connective tissue called the endomysium. And when we pull out one of these fibers from the bundles of fascicles, they're called a muscle fiber or a myofiber. And that is basically the muscle cell. So here we can see its individual nuclei. So that is a muscle cell or a myofiber. Now the myofibers then are made up of bundles. The myofibers are made up of bundles of myofibrils. Okay. And here there are some other interesting features that we can see. We can see the sarcoplasmic reticulum, which is sort of like a casing surrounding the muscle cell, the bundles of myofibrils. And if we pulled out a single filament from the myofibrils, we can see them here. And there's basically two types, actin and myosin. This is the thin, and this is the thick. And the fact that one is considered thick and one is thin gives us these stripes here that make up the sarcomeres. Sarcomeres is just a unit of the myofibrils. Okay? So make sure that you know this organization of the skeletal muscle tissue, and we'll come back to this in a little bit. So before we can go any further, we need to sort of touch on some nervous tissue. And we need to do that to explain actual muscle contraction, because muscle contraction begins with muscle tissue excitation. And to excite that muscle tissue, we need nervous tissue input. So it begins with the somatic motor nervous system here. That's where we're going to start. And I have here a cell body drawn within the central nervous system. And you remember the central nervous system is made up of the brain and the spinal cord. Okay? And our cell body of our neuron, this is a neuron. This extension is an axon of the neuron, and remember, a neuron is a nervous tissue cell. Okay, so this whole thing is considered a cell. The cell body is found within the central nervous system, and this one's going to be within the spinal cord. So the cell body is found within the spinal cord, and it is going to send an electrical impulse down its axon called an action potential. And it's going to enter into the peripheral nervous system to meet up with a muscle. So I have a muscle drawn here, and once it gets close to the muscle, it'll send a chemical message in the form of a neurotransmitter called acetylcholine. Okay, acetylcholine. This is a neurotransmitter. Okay, and it's sending it across this space right here. This space is called the synapse. And it has a specific receptor found on the muscle. This receptor is called a nicotinic acetylcholine receptor, a nicotinic acetylcholine receptor. And it's not just found anywhere on the muscle. It's in a fairly specific place in an extremely folded area of the muscle. It's a super folded area of the skeletal muscle cell membrane called the motor end plate. And together they call this the neuromuscular junction. They call this the neuromuscular junction, where the neuron meets the muscle. Okay, so this is sort of a brief overview of what we're going to be talking about. Alright, now let's get some anatomy of our myofibrils out of the way. Remember, myofibrils are bundles of these myofilaments, and you remember we have thick and thin filaments. The thick filaments are myosin. They're myosin, which are the contractile proteins, and I've drawn them here so that you can see the heads of the myosins. And remember, it's not just one myosin contractile protein. It is a bundle of these myosin contractile proteins. I read something like 200 in a bundle. 200 of these myosin contractile proteins in a bundle. So in this first picture, we have the myosin head folded. And it has two receptors on them. The first receptor, here in red, is the actin binding site, where it is going to bind to actin, the actin filament. And the one in yellow is the ATP binding site. Now think of this as sort of like cocking the hammer of a gun. to move to this position. We still have our same receptors, and this ATP here has now converted into ADP plus P, but it stays within that binding site. This ADP plus P stays within this binding site, and for it to move back, we need ATP again. Okay? So that ADP plus P stays within this binding site. So that's the myosin contractile protein, sort of a diagrammatic view of what it would look like. Now the thin filaments are mostly actin, whereas with myosin it's about 200 of these contractile proteins. The actin, it's about 2 of them, and they're twisted sort of in this double helix arrangement. And there's some other things here. So you see this white line I have, and then we also need to draw some binding sites on the actin. So on every single one of these actin filaments, on these actin proteins, is a binding site for myosin. But it is blocked by this white line here, and this white line is called tropomyosin. So, and what is holding it in place is sort of this gatekeeper called troponin. In order to get this gatekeeper to move out of the way, to allow tropomyosin to move and open up these myosin binding sites so that it can react with the actin binding site here on the myosin heads, we need to talk to this gatekeeper. And the molecule that talks to the gatekeeper is calcium. It has a binding site on troponin. Okay, so calcium has to come in, bind with troponin, and when it does, that gate then opens, tropomyosin can move out of the way, and our myosin binding sites, all of these myosin binding sites are now open and can react with its actin here on these myosin contractile proteins. Okay. Now if we sort of zoom out and take a look at our bundles of myofibrils, or bundles of myofilaments here, they form what are called sarcomeres, and sarcomeres is just a unit of these filaments. A sarcomere is considered from zetas to zetas. So that is one sarcomere. But what is a Z-disc? Well, the Z-disc here in red is where this is actin. Actin binds to. So all these little actin proteins, these actin filaments are stuck to the Z-disc. And then myosin is here in blue, and myosin binds to the M line. So this is an M line, this is an M line, and then here in the middle is an M line. And we have our myosin bonded to the M line, and here's the little heads on the myosin. And you can see that there is some overlap here. Okay, and that's where they're going to react. And whenever they react, then actin is going to move in this direction. Okay. There's some other places that we need to know, and that is the A band, which is this is the A band, which I like to sort of think of it in this direction, or in this way. So if I drew a spaghetti noodle and colored it in different sections, so if this was the dark band, this is the light band, dark, light. This is basically what you would see if you looked at it underneath the microscope. different bands, different light and dark bands. And this is what the thin and thick filaments make up, these light and dark bands. So now, this region right here is made up of only actin. And the actin, the places that it is only actin, is considered the light band, or the I band. So light, I. Light has an I in it, so that's how I remember it is the I band. So the dark band, let me redraw this. The dark band is the other portion, the dark band. And then the H zone is the place where it is only myosin. So this is the H zone. Okay. Now this page in your notes just reviews the muscle tissue organization. Remember, we started out with the skeletal muscle organ. An example of that was the pectoralis muscle, or the deltoid. And that skeletal muscle organ is made up of fascicles, bundles of fibers, muscle fibers, and then those bundles of myofibers, which is basically the muscle cell, are made up of myofibrils, and those myofibrils are the sarcomeres, and those sarcomeres are stacked end to end to make those different colored bands. And the sarcomeres are made up of the myofilaments, of which we have thick and thin. And don't forget to write down or to memorize the different connective tissues. So remember, you have 3 of these, and remember where they're at. Now I get to talk about skeletal muscle excitation, and remember, this happens where the nerve actually meets the muscle, called the neuromuscular junction. So here we have our somatic motor neuron, and remember, we're going to send an action potential, which is an electrical charge, down the axon of the neuron, and once it reaches the terminal of the axon, it'll release its neurotransmitter across that space. Remember, that space is called the synapse. And communicate with the muscle. So let's look at that in a little bit more detail. So here we have our action potential traveling down towards the end of the neuron. And remember, this is an electrical charge, so it holds a charge. And since this is an actual cell, it holds a resting membrane potential. So whenever you're changing the charges of something, it's going to change the membrane potential. And here we have that membrane potential as being negative. When the action potential travels down, it becomes more positive. And by doing this, it opens up a gated channel. This gated channel allows an ion to flow through, called calcium. Calcium comes in, binds with its receptors on these little vesicles held within the end of the axon terminal, that hold the neurotransmitter. That neurotransmitter is exocytosed out across the synapse till it finds its receptor on the muscle. Remember, that specific place on the muscle is called the motor end plate. Remember, these receptors are called nicotinic receptors, nicotinic acetylcholine receptors. And these nicotinic acetylcholine receptors are basically sodium channels. And by binding this acetylcholine with the nicotinic receptor, it allows sodium then to flow in the muscle at the motor end plate. Okay, that is excitation at the neuromuscular junction. And once we get sodium inside, it changes the membrane potential of the cell. And I don't really want to call it an action potential, because it really isn't an action potential. It's not. This isn't a nerve cell. It's muscle tissue. So we're going to call it a depolarization wave. a depolarization wave. So here we're having sodium flow into the muscle, and since we're adding this positive charge to a cell that was, had a resting membrane potential of negative 70 millivolts, it's going to cause that resting membrane potential to become more positive. And it sends this depolarization wave across the muscle cell. And the steps after that, that's what we're going to look at next. Okay, so hopefully, this is drawn a little bit differently, but hopefully this looks familiar. We have our somatic motor neuron. The active potential is traveling down towards the terminal. That changes the membrane potential of our neuron. Calcium flows in, finds its receptor on these little vesicles that hold acetylcholine. Acetylcholine gets released out to the synapse. It finds its receptor, its nicotinic receptor, on the motor end plate, and that triggers sodium to come into our muscle cell. And once it does, that changes the membrane potential of our muscle cell, and it causes a depolarization wave across our muscle cell, meaning it changes the membrane potential of our muscle cell. And it travels down this special structure. The special structure is called a T-tubule. It helps to distribute this depolarization wave. And there's something else within this muscle cell, the sarcoplasmic reticulum. Our sarcoplasmic reticulum is basically a storage for calcium. It has a high concentration of calcium. And it has this high concentration of calcium because of these active, these active calcium transporters. Meaning it is constantly and actively, meaning it requires ATP, pulling calcium out of the sarcoplasm, the cytoplasm of the muscle cell, into the sarcoplasmic reticulum. So it keeps this concentration high. Okay, so now, going back to our steps, sodium came into the cell, we had this depolarization wave, it traveled down our T-tubule, and it opened these voltage-gated calcium channels. Okay? And then calcium that was held in our sarcoplasmic reticulum was allowed to go down its concentration gradient out into the muscle cell. Okay, the reason it went down its concentration gradient is because, remember, it was in high concentrations in our sarcoplasmic reticulum, thanks to this active transporter here. Okay, so now we have calcium that's going down its concentration gradient into the muscle cell. And this is where number 9 starts, where we actually get muscle contraction. So from here, calcium will bind to troponin. Remember the gatekeeper? Calcium will bind to troponin, and it moves tropomyosin out of the way, and the myosin binding site is revealed. Then myosin can bind to actin. Okay, myosin can then bind to actin. Then this, that was held within this site, because remember this is in the cocked position, this right here gets kicked off, and this whole thing moves, or in other words, it's bent again. So we get contraction. It gets contraction, and what is back in this place is its place right here, ATP. So that this will move back into the cocked position. Okay? So ATP, we'll see, needs to be here in order for the myosin head to relax, or for the muscle to relax, which we'll see here in a second. Okay? So, just to review relaxation, myosin must go back into the cocked position And to do this, it needs ATP. Okay? So we're just working in reverse here. Calcium is actively pulled back into the sarcoplasmic reticulum. Sodium moves back through its special transporters. Let me erase this. I think that's supposed to be acetylcholine. So sodium's going to move back acetylcholine from the nicotinic receptors. Hold on. I got ahead of myself. I'm sorry, guys. So calcium is actively pulled back into the sarcoplasmic reticulum, and this causes the removal of calcium from troponin, and then this causes tropomyosin to move back over actin. Or I might say in your notes, the myosin binding sites. Either one's fine. Okay? So this whole time, this action potential stops. Acetylcholine unbinds, and sodium, here's where sodium is. Sodium moves back out through those special transporters. Remember, those were the nicotinic transporters. Acetylcholine is still out in the synapse. We need this special enzyme. This enzyme is called acetylcholine or acetylcholinesterase. I know that's a mouthful. Remember, if it ends in ASE, it's an enzyme. So acetylcholinesterase. It's an enzyme that breaks down acetylcholine into acetate and choline. Okay, so make sure you know this. Now that we've talked about how we actually or physiologically contract muscle, where does the energy come from? Basically, there are three ways. It's either really quick, short-term, or long-term energy. Now, if it's really quick, I'm meaning like 10 to 15 seconds, and it relies on the ATP that is inherently within the muscles. Okay, and it's called the phosphagen system. And with the phosphagen system, it takes ATP that is inherently in the muscles and creatine. Okay, and it uses that ATP, and when we use ATP, we make ADP and creatine phosphate. And to convert that back, or this is just the way the system works, as it converts it back, it needs an enzyme called creatine kinase. To restore it to ATP and creatine. Okay, it transfers a phosphate to the ADP to make that ATP the energy that is going to be used to fuel this 10 to 15 seconds of fast energy. Okay, so this does not last very long. It is not very efficient. It's just going to use the ATP that is inherently already there. It's not pulling out any other sources of energy. And it requires this enzyme creatine kinase to convert it to ATP and then back into creatine and stored it in the muscles. Okay? Alright, so now if we're looking for a little bit longer So that's 30 seconds to 1 minute. Short-term energy, it's going to depend on your blood sugar, your blood glucose, and the amount of glycogen you have left in your muscles. And glycogen, I like to think of glycogen as sort of your body's way or animal's way of storing glucose. So you know what starch is? Starch, for instance, is found in potatoes. Starch is basically long chains of glucose molecules. Remember, glucose is a monosaccharide. It's the form of the carbohydrate that we need for cellular respiration. But if we're not using that glucose, then it gets converted into something in the body. And in animals, it's converted into glycogen for a short amount of time. Okay, in plants, it is converted into starch. Alright, so in anaerobic, which is short-term energy, anaerobic, we do not use oxygen. So if it is anaerobic, there's no oxygen in this metabolism. So, for instance, if we were to use glucose, glucose goes through a process called glycolysis. And we end up making 2 ATP and 2 pyruvate from 1 molecule of glucose. So that's not much energy. It's only 2 ATP. And we also have a byproduct of lactic acid that gets dumped into the blood. Okay, so here we don't use oxygen, and we end up making this byproduct here. So now if we really want to have a lot of energy, we have to use aerobic cellular respiration, which requires oxygen. So this is slow but efficient. A lot more efficient than these other 2 ways. So let's look at this on another page. So aerobic cellular respiration. It begins a lot like anaerobic. We start off with, say, a molecule of glucose, and it goes through glycolysis. And of course we end up making 2 ATP and also pyruvate. And then that pyruvate goes through a process called the citric acid cycle, or the Krebs cycle. Same thing. And we make these electron carriers. We make a few ATP, and then we end up making these electron carrier molecules, these high energy molecules. And then those electron carriers go to the electron transport chain. And here's where we make the most ATP. But to do all this, we need oxygen, because oxygen ends up being the final electron acceptor here. This is important. So we end up making lots of ATP, something like 26 to 28 ATP from the electron transport chain. And then our byproducts are water and CO2. And what also is cool about this is that if we didn't have a molecule of glucose, we could end up using fatty acids or even amino acids, units of proteins, units that make up proteins. Okay. So oxygen debt is another term that we need to be familiar with. It's basically the oxygen needed to restore pre-exercise conditions. So how much oxygen we had before we started to work out or exercise or exert energy. Oxygen, we know, needs to be replaced. We breathe because oxygen needs to be replaced on hemoglobin, Remember, hemoglobin is that protein in your blood that carries oxygen. But we also breathe not only to replace oxygen on hemoglobin and then in turn feed our cellular processes, like through cellular respiration, but also to replace oxygen on hemoglobin's counterpart in the muscle called myoglobin. It's basically the hemoglobin in the muscle. It carries the oxygen in the muscle. Also, to restore this oxygen debt, our glycogen stores need to be replaced. The ATP crenate phosphate phosphagen system needs to be replenished. And the lactate needs to be converted back to glucose. Okay. Now I want to talk about muscle fatigue before we go on to the types of muscle fibers that we have. What is muscle fatigue? It's when, the muscles can't sustain the expected power output. And there are basically two types. One is central and one is peripheral, meaning one comes from the central nervous system and one affects the periphery of our nervous system. If it is central, they term this mental. And they term this mental because it is basically subjective. And if something is subjective, it can't really be quantified or counted. In some ways, this is considered protective. So, for instance, if you were told to continually run up a hill, your brain would say, God, do I really need to run up that hill one more time? That is your central nervous system's component of fatigue. That's the mental version, and you can see how it's your body trying to protect the rest of you from getting hurt. And for this reason, it comes first before the peripheral version of fatigue. So there are some examples I have of peripheral muscle fatigue. So that is, I have this little diagram here. Peripheral starts from this portion all the way down. So that's your somatic motor neuron from that action potential reaching there and then all the way down through the muscle. So things that might contribute to peripheral fatigue are a decrease in the neurotransmitter acetylcholine, our decrease in our energy supplies, so like ATP or glucose, the things that fuel cellular respiration, a decrease in our glycogen stores, or a decrease in even oxygen. This equation here, ATP, it's converted to ADP plus a phosphate. Well, they think that as we continually use that muscle, this phosphate right here will sometimes bind with calcium, taking it away from the muscle. So if you remember our gatekeeper that had a binding site for calcium, that gatekeeper was troponin. That required calcium to bind with it before it moved out of the way and allowed tropomyosin to move out of the way from the myosin binding sites on actin. So they think that could possibly contribute to muscle fatigue. Another one is potassium in the extracellular fluid. They think potassium gets out of those nicotinic receptors, changing the membrane potential. And then the last one is an increase in lactic acid might actually change our body's pH. So all these we can see are physiological, while this is particularly psychological. Now we can take a look at skeletal muscle fibers and compare them. I know your book gives you more than just two types, and that's fine. I think they're different enough that we can just do two types that, and give us that there's a broad enough range that we can just do a comparison between the two broadest types, the two types that are farthest apart. And those are just generally slow twitch and then generally fast twitch. Those are the ones that I'm going to test you on anyways. Okay. So the difference between the two is, well, basically the speed. In fast twitch, they can contract 2 to 3 times faster than slow twitch. And a lot of it has to do with the type of myosin they have. So they can have what's called fast myosin, which can actually take calcium out and back into the sarcoplasmic reticulum faster. And some of it has to do with this enzyme called ATPase. It's an enzyme that splits ATP. Okay. Slow twitch, these are aerobic. These are generally aerobic, whereas fast twitch is anaerobic. So that might actually give you a clue about fatigue. Which of these do you think would fatigue faster? Well, hopefully you said fast twitch. Now, the diameter of these muscle fibers will tell you something about power. Fast twitch actually have a bigger diameter. They have a bigger diameter, and the bigger the diameter, the more powerful they are. The bigger the diameter, the more powerful they are. Talking again about cellular respiration, slow twitch fibers have a higher concentration of myoglobin. And if you remember, myoglobin is basically muscle's version of hemoglobin. It likes O2, so it helps to store O2 within the muscle. They also have a higher concentration of mitochondria, which makes sense because these are the ones that are doing the most cellular respiration. Okay? So make sure you know these general comparisons of muscle fibers and also these terms. The next term that you need to know is twitch. A muscle twitch is one message, an action potential followed by the shortening of the muscle. So I'm guessing that these graphs look familiar to you. You did these in lab. Hopefully you did these in lab. What I want you to know from this is what summation is, what tension is, what tetanus is. Okay. And just so that we're clear, make sure you know what summation is. That's by adding another stimulus before the muscle is completely relaxed. You increase the tension. That is summation. This doesn't work with neurons. Action potential is an all or nothing type of thing. This only works with muscle cells. So you can only have summation with muscle cells. So make sure that you know those definitions. Now with motor units, there are some rules that we have to follow. Motor units are one somatic motor neuron, and all the myofibers it innervates. So I have here one somatic motor neuron, and then these are the 4 myofibers or muscle cells that it innervates. And it could be 4, or it could be 4,000 muscle cells, and that could be considered one motor unit. The size of the motor unit varies depending on the muscle group. However, all the muscle fibers in a motor unit have to be the same type. So for instance, okay, here we have one somatic motor neuron. It's going to send its action potential to all of these because it's an all or none. But all of these muscle fibers have to be either all fast twitch or all slow twitch. They can't be just fast twitch, slow twitch, fast twitch, slow twitch. They all have to be the same fiber type. Motor units, remember, are stimulated in an all or none fashion. So I can't just stimulate like half of these and then the other half. It's all at once. Okay. Now let's do sort of an example here with contraction force. So I have here 4 motor units. These are the somatic motor neurons that are innervating their muscle fibers. So let's say with our first one, this one will have 10 muscle fibers. The next one will have 20. This one will have 30. And say this one will have 40. So they're different sizes. Now we need to decide what fiber types they're going to be. Let's say that this one is fast twitch, this one's slow twitch, this one's fast twitch, and this one's slow twitch. Okay? The CNS, the central nervous system, can control how fast the action potential is sent, or which motor units to activate. So, for instance, it can say the action potential needs to go this fast, or the action potential needs to go this fast. Either way, the central nervous system gets to say this, or it gets to say, I want to activate 1 and 2. Okay? Alright. It does this thing called asynchronous recruitment. So make sure that you know this word. It's all motor units that are affecting a certain muscle are going to fire at different times. So asynchronous recruitment is all motor units affecting a certain muscle are going to fire at different times. So why might this be beneficial? It's because when one is not firing, the other is resting. This reduces the chance of what? So let me ask you this again. So, because when one is not firing, the other is resting. When one is firing, the other is resting. This would reduce the chances of what? Fatigue. So it's like saying, I want to fire all 3 of these and then rest this one, or vice versa. And by doing this, it reduces the chances of that muscle group fatiguing. Okay? So make sure you know what asynchronous recruitment can do, and also that the central nervous system can control the type, the number of somatic motor neurons, and how quickly the action potential is sent. This can actually change the kind of contraction that happens. Okay, so we only have 2 more slides to talk about, and then I want to talk about some specific types of contractions and then some effects that exercise can have on muscles. So the 2 types of contractions I want to talk about are isometric and then isotonic. With isometric, there is no movement. It is muscle contraction, but there is no movement. It is due to the elastic quality of the sarcomeres. It happens because, or for instance, if you imagine pushing against the wall of a building, there is no successful muscle shortening. It really only happens if the load is too heavy. Okay? So it is muscle contraction, but there is no movement. Whereas with isotonic, it is the same force all the way through. It is very smooth, and we have 2 types. It is either concentric or eccentric. So with concentric, there is actual shortening of the muscle. With eccentric, it lengthens the muscle. So for an example of this, picture holding your arm out with something in your hand. The bicep is still contracting some, but it is still lengthening at the same time. So just picture holding your arm out with something heavy in your hand. The bicep is still contracting somewhat, but the muscle itself is actually lengthening. So that is eccentric contraction. So know the difference between the two. Now on this last page, I want to talk about some of the effects exercise actually has on muscles. Hypertrophy is when the muscle cells actually enlarge. You get an increase in myofibrils and myofilaments. You get an increase in the mitochondria and an increase in glycogen reserves. It actually increases the ability of those muscle cells to produce ATP. Then the one at the bottom, I think your notes have this wrong. It should say hypertrophy, HPO, HYPO, or atrophy. This is a decrease in muscle cell size. Okay, this is due to disuse and non-exercise, so it is the opposite of this, hypertrophy. The other one is called hyperplasia. This is an actual increase in the number of skeletal muscle cells. This is a disputed issue, and it's sort of unclear whether or not this actually happens. So this is an actual increase in the number of skeletal muscle cells. These are just talking about the size of, these two are just talking about the size of the muscle cells. This is talking about the actual number of skeletal muscle cells. Okay, so that concludes our video for Chapter 10. Thank you. Happy studying.